Hello, everybody. All right, I'm going to start this week's lab. So let me just change my share. This is the muscle anatomy. Oh, of course, it's not connecting. So hold on. Let me go back into that page. There we go. There's that. Hold on there. Oh, that's how I'm going to do it. Okay, hold on. There we go. Now, let me try to share this. Let's see. Nope, still doesn't want to do it. Hold on. I want to share to that. Let's hope that gets it. It's just not connecting to that document. So let's try doing this. We'll just go through it this way. Yep. All right, we'll just do it that way. I don't know. Lately, it always seems like something with this Zoom and whatnot. All right. So we are working on Lab A Muscular Tissue and the System, Part 1. We're looking at some slides and some models kind of here on the first page. So we have a slide of dense regular connective tissue, and what is its function. So when we get to that and we look at the slide that's on, is in the image base here on the course page, it has generally a peachy kind of appearance, very parallel fibers. So if you saw that slide, you'd say identify that tissue, you would say dense regular connective tissue. Function, this is the tissue that forms tendons and ligaments. Tendons attach muscle to bone, ligaments attach bone to bone. Okay, then we're going to look at skeletal muscle anatomy. Right, we look at a model first, which is connective tissue layers of a skeletal muscle model. Those are the epimysium, the paramysium, and the endomysiums. So the epimysium describe a function. You could say surrounds entire muscle, forms tendons, right? Because the epi is above and around, surrounds the entire muscle and forms tendons. Paramysium, connective tissue surrounding one fascicle. And the endomysium is connective tissue surrounding one cell or one fiber. Right, so we have connective tissue surrounding one cell or fiber that essentially connects, interconnected to the connective tissue paramysium that surrounds one group of fibers into one fascicle. And then all the fascicles get joined together as an epimysium and then forms tendons that attach that muscle to two bones. All right, then we had the skeletal muscle slide, just function. I'd say contract and produce movement. That would be fine for function. Generate heat. And then we look at a neuromuscular junction histological slide. I had that in the notes already, where it's either like a grayish background generally, or red is a common one. So you have the muscle tissue in the back, and you have a neuron on the outer surface. It looks like a black spider web with little tufted ends. So you're to identify which part of the skeletal muscle, maybe which part is the motor neuron axon and which is the axon terminal. I believe on the image that Dr. Brightman has up there, it only points to the axon terminal, but I'm going to point to the motor neuron axons as well. They're just the black lines, that's all. And then we have muscle physiology. All right, so the motor neuron and axon terminal, sometimes you can see a little bit of this acetylcholine, not always on the models. You can definitely see transverse tubules, sarcoplasmic reticulum, actin-myosin. So it says identify, in number 4, muscle physiology, identify the following structures on all muscle fiber models, all muscle fiber models, which means it's going to be helpful to come to class and describe functions, which we're going to talk about the functions right here. Right? Structures here are in correct order of muscle stimulation. So what does that mean? In lecture, we specifically talked about the sequence of events, starting with an impulse traveling down a motor neuron to where you get a power stroke and you have force generated. Right? So these structures here, it will go in sequence. First, you have a motor neuron, then the axon terminal, the acetylcholine that's released, the sarcolemma to the transverse tubule on top of the sarcoplasmic reticulum, and the roles of actin and myosin, right, which are the thin and thick filaments. So that is the order, given these structures, that they would be in order from where we start to generate an impulse to where we have some kind of contraction occurring. So next to that would be your matching, and then you can, if it's not brief enough discrimination, if it's not a brief enough definition here, then you can write out something that synopsizes that a little bit and fit it in this function column, just like we've been doing before. Right now, it's not that imperative that you do that because these are really true, false, multiple choice questions because we're doing this online, but face-to-face classes, that's about the space you'd have to write out your answer, in 45 seconds or something, so that's why we try to train you into, writing out and finding a response to, let's say, the function of an axon terminal that would fit on this appropriated or available space on an answer sheet that would be provided for you in a face-to-face class. But yours are going to be pretty much true, false, or occasionally a fill in the blank if you're so lucky, but most of them are fill in the blank and true and false, something like that. Okay, so let's do these matching. So I always suggest that you look at the A, B, C, D, E, F, G functions, and then you match it to the term. It goes faster that way. So the thin filament with a binding site for cross-bridge formation of myosin, right, has a troponin tropomyosin complex on it, right? The thin filament is actin, all right? So A goes in here for actin. Then we have B, the thick filament that binds to actin and produces your power stroke. So the thick filament is myosin. Then we have C, the neurotransmitter that binds to a receptor on the sarcolemma. That would be acetylcholine. D carries impulse to the deepest region of the cell. That would be your transverse tubule. E contains acetylcholine receptor sites. Well, the receptors are only found on the outer surface of a cell, right? So that would be the sarcolemma. F stores and regulates calcium ion release. That would be the sarcoplasmic reticulum. G carries the stimulation away from the spinal cord. That is your motor neuron. And then has or contains vesicles that release acetylcholine when stimulated. That would be the axon terminal. Right. So you should know the sequence of events beginning with an impulse traveling down this motor neuron, and then what happens at the axon terminal, because we just identified their functions. The acetylcholine binds to the sarcolemma, right? The sarcolemma has receptor sites for acetylcholine, essentially creates another impulse or potential, action potential. It travels down the transverse tubules to the interior region of the cell. That stimulates the sarcoplasmic reticulum to release the calcium it stores. Calcium essentially is going to bind to troponin tropomyosin, unblock binding sites on the actin for myosin to form a cross bridge. And then we have a power stroke and we generate force in the cell. Right, so that's that check. What we're going to do is then kind of apply take those functions that we've just identified here, and we're going to apply them to any structure we can see on the models. If it represents the motor neuron axon, an axon terminal, acetylcholine, these are the functions you can use. Okay, then that takes us to our first models. Yay. So that's our, physiology, some of it, the physiology and the microanatomy of the muscle tissue. And now we're just going to be identifying certain muscles listed here. Here we go, head and neck muscles. And here's a list. You need to be able to identify all these muscles on the models: orbicularis oculi, orbicularis orbis, zygomaticus, mylohyoid, blah, blah, blah. All right. If you need to know the origin or insertion or the action, I filled it in for you. All right. So that makes it easy. I'm not making you know all the origins or the insertions or the actions, because for some of these, they're circular muscle anyway. But some of these are rather easy actions, to commit to memory. They're fairly basic to let, if you just look at where the muscle, where it begins and where it ends, what's its origin, what's the insertion, and when it shortens, you can kind of see what effect it's going to have on the bone. So it's maybe less than half of all the information you need to know. So I filled it in for your head and neck muscles, and then we're going to move on to dorsal muscles, and I've done the same thing. You need to identify all your muscles. If I want you to know an origin insertion, I've given you one. Or if I want you to know the action, I've given it to you. If there's a dotted line, then you don't have to know them. All right, I'm not going to ask you them. I believe we have one that shows the erector spinae. If there is not an erector, I think there is, but if there isn't an erector spinae on one of the images there, I do have some videos where I go through all these models. So, use both of those now. I said that once I got to the muscles last spring, this is about the time, when we had spring break, we essentially never came back from spring break last spring. I could get into my office for a few days, but that was it. So for all these labs going forward, I did a video and I put them up on YouTube, and that's what this goes to. It should direct you to these other YouTube links. And I think in the video it says I might not ask about slides, but I can't be held to that now, because now they want us to ask some slides. I don't know if I did actually. I probably, if there's one out of this group, it's probably a lot. Okay, so that's really it for filling in the document. All right, so I'm going to close this down. I'm going to stop sharing with that, and I'm going to get into, let me shrink that. I'm going to get into the rest of the course page so I can now see the images. So let me go back to course content and go to Muscle Lab 1. See, so here we have Muscle Lab, when you go to the course content, you go to course material for exam 2. Lab 8 is muscular tissues and system part 1. Immediately below that is the PDF document, this document. So print that off or save it and, fill in all your blanks. Right. Below that, you see where it has that link. Right. That's the video I did last year and uploaded. Right. But now I'm going to go through the images. Right. So we're going to go, and then below that is the quiz. All right. So here is lab 1. Let me do my share. Okay. So the first image, lab 8-1, that is your skeletal muscle. Most of the time, those slides are in these grays, and I know it's a little vague to say, but you get the nature of these very parallel fibers. Almost looks like the collagen fibers of fibrocartilage that are very wavy, but some portions of it are rather straight and linear, and some of them are a little wavy because the tissue is relaxed. All right, but very parallel fibers and multinucleated. That tissue is skeletal muscle. Function, you can say something like to contract and shorten, maybe generate heat, something like that would be fine. All right. Here, Dr. Bartman's told you, yes, it's skeletal muscle. Some unique characteristics is that it's voluntary and multinucleated, but that doesn't really tell you a function. The functions are what I just went over in the previous section. They contract and generate force. They produce heat. Give me something like that. Okay, then this is a little difficult to see, but it does have a pinkish and orangey kind of hue to it. In class, if you can come and see them in class, you'll get a much better idea of what it looks like, but it's generally in like this orangey, peachy, maybe a light pink tissue that's very parallel like this. This is dense regular connective tissue. Right? You don't see nuclei, so it's not skeletal muscle, and you don't see any striations. See, that would be dense regular connective tissue, and the function, can be forms, tissue, forms tendons and ligaments. So tendons attach muscle to bone, ligaments attach bone to bone. Let me go back to pictures and just so you can see what I was just referring to. We're looking at the muscle, and you can see those striations, those parallel lines that are perpendicular to the length of the muscle. So those little, like you took a pencil and drew little parallel lines, those are striations, and you can see all these nuclei scattered here to the periphery and within the muscle cell. Right, so now you know that's a muscle, that's muscle tissue. Right? But dense regular connective tissue does not have striations. Yes, it's parallel and kind of wavy-ish, but no striations, no nuclei that you can see. All right. Okay. Now we'll go on to the next image. This is our neuromuscular junction. All right, so the background, this slide just happens to be in purple, but the most of them in the lab room are going to be a red background. And look, you can see striations. That is skeletal muscle tissue in the background. In this case, it's purple, and some of the other slides, it's going to be red. Right. The black lines with the little balls at the ends, those are the motor neurons. Right. So the black lines are the motor neuron axons. And when you get to those little circular structures, those are the ends, that terminate in the axon terminals. Right. So the ball areas in black, there are axon terminals. And if you can magnify in lab, and usually on the overhead projector that I'll zoom on Thursday, we'll show this as well. If I magnify these, you will actually see the little synaptic vesicles in there. They look like little water balloons and a big water balloon kind of here. So you can actually see the synaptic vesicles that store the neurotransmitter. Right, so you have a neuromuscular junction. You have axon terminals. Those are at the ends. The skeletal muscle in the background. But the black wiring here, those are motor neuron axons. Okay, then we get into these models, and this is where it gets a little more difficult, just trying to do this, flat like this without actually seeing the model. Because when you see the model, you can turn it around and stuff. You're like, okay, from once I can see how, like this structure over here, 1 and 2, these are parts of your motor neuron. To the left, in the different reds and green here, the blue, the light peach, that's all within the muscle cell. So 1, this is pointing to the motor neuron axon or the motor neuron. And then 2, you can see those little white balls, right? Those are, here we are in an axon terminal, and those little white balls would be the synaptic vesicles stored there. And those other ones, those are just some mitochondria, like little red jelly beans, right? Because it has some normal cellular anatomy, organelles, in the axon terminals and the axon. But then here at number 3 is where the neuron meets the muscle cell. So 3 is pointing to like the light peachy kinked kind of region here. And that continues actually to the rest of the cell. Now, if you're not looking at number 3, just look to the bottom. You see those blue tubes, and then you see the light peach, pink, like pink, really, outer membrane here. That is both sarcolemma. Right, so I generally point to the outer pink part, which is this more flatter pink region called the sarcolemma. But where it kinks up on itself, at where the number 3 arrow is pointing to, it's the same thing, right? It just kinks inward a little bit at those sites where you have a neuromuscular junction. That's all. Right, so we'll start by just identifying 1, 2, and 3. 1 represents the motor neuron. 2 are the balls, those white balls, the synaptic vesicles that store acetylcholine, or your neurotransmitter, which is acetylcholine in this case. And then 3 is pointing to the sarcolemma, the plasma membrane of a muscle cell. Okay, so same model, all right? Now we're just pointing to different things. So personally, again, the sarcolemma, here, number 1 is pointing to the light peach network, and 2 is pointing to those blue tubes. But if you go to the bottom of this model, I think this is easier to kind of picture. That darker pink outer membrane is the sarcolemma, right? This is the plasma membrane of the muscle cell. It's also there at number 3 of the previous image, but you can see here the sarcolemma pinches inward, especially that part right in the middle. You can see it pinches inward, and now that the outer portion of it is blue, but the interior is still that light pink, darker peach region. That's because the transverse tubules in blue are an extension of the sarcolemma, its plasma membrane. And that gives you kind of a visual of like, oh yeah, it pinches inward. And when the impulse is generated, an exponential is on the surface of the sarcolemma, it travels through the interior of that tube. Right? And the outer portion, is colored blue, so you can distinguish it from other structures. That's all. Right? All right, so the blue tubes are the transverse tubules. 1 is the sarcoplasmic reticulum. And so you should be able to, given the structures that, or the functions of all those different structures in sequence, the motor neuron, the axon terminals, the synaptic vesicles, blah, 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 sarcolemma, transverse tubule. We just covered that. It was what, exercise 4 or something. I should be able to point to a transverse tubule and you tell me what the function is, or a sarcoplasmic reticulum, tell me what the function is. Actin or myosin, which is what's identified here, or the motor neuron, to the first image of this model that we just looked at. Remember, we're going to use the functions from the lessons we filled out in our paperwork, and we're applying it to these models. Even if the next card doesn't tell you that, that's what you're doing. Okay, so other things that you can identify here, the little skinny red lines versus the darker, chubbier red lines. Right? Skinny actin filaments, they're the fin filaments, and the myosin are the darker, thicker filaments. All right. So remember, actin is the molecule that has troponin and tropomyosin blocking it. Myosin has like the golf heads, that want to make contact with actin and row toward the center and do the power stroke, form a cross bridge, then power stroke, and get that muscle cell to shorten. Now, this is a different type of model, but it's showing you connective tissue to the outside here. Number 1, that would be the endomysium. 2 is the plasma membrane of the sarcolemma of the muscle cell. See, and it's just a different type of model, but it's kind of showing you the same thing. Here's a motor neuron, right? Terminating here the axon terminals, the little, distal ends, or the little bulbs, right? They cut one of those axon terminals so you could see what the receptor region for acetylcholine would be on the sarcolemma. It's a slightly different shape. All right, so we have these axon terminals here, and here we have receptor region on the sarcolemma. 2 is just pointing to generally the sarcolemma. We're going to get to those other ones in a second. I get a little excited and ahead of myself. So 1 is the endomysium, and 2 is the sarcolemma. Okay, then we're looking at the other structures. So here we have a motor neuron. In the other model, they cut it open, so you're looking to the interior of that motor neuron. Now, after lecture, hopefully you get that this is going to be myelin sheath covering the axon that's in the center. Here we have our axon terminals. We have a receptor region on the sarcolemma. And so motor axon, axon terminal, and a receptor area or site on the sarcolemma. This is another type of model. There are a few different ones. I don't use this very frequently, except for maybe the sarcoplasmic reticulum in blue and the green transverse tubules that sit on top of it. Because it's just kind of old, and I like the other one better. But 1 and 2 are pointing to the actin and myosin filaments. So this is a great big model of a muscle cell, right? So 1 is the myosin, 2 is the actin. Thick filament versus thin filament. This is the model I like better because the gray stands out for me visually better. I don't know, maybe I need new glasses. Right? But number 1 is pointing to the transverse tubules. In that first model we looked at, the transverse tubules, they made them blue on the outside. Now they're green. And then instead of having light peach network, of sarcoplasmic reticulum, these are yellow. So they're just more brightly colored, that's all. And then down here in the grays, you see where you have chubby black gray and then thin grays at the end, right? The chubbier dark gray would be the myosin, and the thinner dark gray, would be the actin. And the light gray in between all those and in the background here, that's just the cytoplasm of the cell. That's why it's continuous light gray all over. But when you have the big green lines, the yellow sacs all interconnected. And then when you look to the bottom of this model, the thicker, especially right through the midline there, the thick dark grays versus the skinnier, thinner dark grays to the right, that represents the difference between myosin, that's the thick, that forms cross bridges with actin, and then the thin actin filaments with binding sites for myosin. Okay, so primarily the 1 and 2, really just trying to show those two structures, but I wanted you to see the other examples of actin and myosin. So transverse tubules and sarcoplasmic reticulum. Transverse tubules carries impulses to the interior of the cell that are generated on the sarcolemma, and you can kind of say whatever you want. The sarcoplasmic reticulum, stores and releases calcium. Remember, it has to be stimulated to do so. Okay, then we're getting into our muscles, right, on our different models. And again, a lot of these, it's a lot more fun to see them on the models in lab because we either have torsos that are about human size, that are just on top of the table, but we have these great big muscle men, these big models in the room. And so you start to see the location and size that you would find or expect to find them on a human being, not just on a piece of paper. So, and I know these are kind of flipped sideways, but this is identifying the circular muscle around the eye. Here's the circular muscle around the mouth, and this one in the neck area to the anterior aspect of your neck. So you first start by just identifying the muscles, and if you did nothing else with all these, I would just be able to name every muscle. And then I would go back and go, okay, with my little chart, which one is the first origin, insertion, or maybe action I have to know, and then go back and do your origins and insertions and actions. So number 1 was orbicularis oculi, the circular muscle of the eye that you do have to know in action. That is your winking eye, or closes the eye. You wink or close your eye. Orbicularis oris, circular muscle of the mouth, closes the mouth or purses the lips like you're going to kiss, or you need to close your mouth because you're eating something and you don't want food to fall out. And then you have sternocleidomastoid, right, named for the 2 origins and 1 insertion, sternum, clavicle, and mastoid process. That muscle, when it contracts both sides, helps you nod your head forward like yes, so it brings your chin to your chest. If one side contracts, it kind of rotates your chin to the opposite shoulder and kind of brings your ear down toward your chest. Kind of a weird little function. You do have to know these, especially sternocleidomastoid and trapezius, because they are innervated by a certain cranial nerve of the facial muscles. There's certain ones that we're going to get again when we look at our cranial nerves, because they stimulate or provide the nerve motor supply to muscles that are associated with facial expression, like raising your eyebrows, or they supply muscles that are associated with chewing muscles, like, the masseter, things like that. Okay, then we look to the side of the head, and here we see more muscles. 1, 2, 3. Right? One is temporalis, pertaining to the temporal bone. Right? We have the masseter, right? And then we have zygomaticus. There's a zygomaticus minor and major. We're just gonna group it together as zygomaticus. All right. So here's temporalis. It originates on the temporal bone and inserts on the mandible. Here we have the masseter. Right. By the way, let me just finish this. The masseter really originates on the maxilla, which is the upper bone of the mouth, and inserts on the mandible down here. Both of those close the jaw. So you have to open your mouth to put food in it. These will contract and elevate your mandible, close your mouth so you can chew. So they're chewing muscles. And then 3 is pointing to zygomaticus, named for its origin on the zygomatic bone, to the corner of the mouth here, or orbicularis oris. You can use either answer. And that is a smiling muscle. So when that contracts, it elevates the corners of your mouth and you get smiling. Temporalis, masseter, zygomaticus. Remember, you need to know the specific origin, insertions, and actions that are on the chart that I provided you. Okay, then we're looking at the undersurface of the chin, right? So especially when there's certain models that where the head can come off the rest of it, and so you can lay it there, and you can easily see the muscles under the chin, right? And there's 2 you need to know: a digastric and a mylohyoid. 2 is pointing to a mylohyoid. These also to the side, are mylohyoids as well, because you can tell by the ones on the side are the better example of the mylohyoids. But you have a mylohyoid and a digastric. They both close. They help close, open the mouth when you want to open it, and then the temporalis and the masseter will close the mouth. 1 is the digastric, and 2 is the mylohyoid. So when they contract, they're going to pull your chin downward. So you open your mouth, and then those other 2 muscles, temporalis and masseter, close it. The masseter muscle, I call it the pit bull muscle, because those are the muscles that are really strong and huge on pit bulls. They have this tremendous bite pressure, bite force that they can produce. Okay, then down here, we're looking at the chest. And here we're looking at serratus anterior, I believe. He's using that as serratus anterior. Serratus anterior. Then we have on the back side, the head is to the right, his butt is to the left, it's sideways. I don't know, again, why these keep coming out sideways. But in any event, you can always rotate them when you look at them, or you can print off some of these images and just make flashcards. 1, right? You have muscle 1, muscle 2, and muscle 3. We have a splenius muscle here at 1. We have levator scapulae at 2, and a rhomboid. We technically have a rhomboid minor and a rhomboid major here, but we put them together like the zygomaticus, and we just say it's a rhomboid or rhomboideus muscle. Splenius, levator scapulae, and rhomboids as a group. Okay, then we're going to look at more of the back. So, when it comes to models, and again, in classes it's easier to show you, but whenever it comes to these models, you have one side that shows you the superficial muscles, the first layer of muscles you're going to see when you peel someone's skin off. The other side will show you the muscles that you can see when you pull off the first layer of muscles, because there's about 4 layers of muscles easily, without even getting into the teeny weeny ones, 4 layers of muscles of the back here. So the most superficial ones, right, is a big muscle of the upper back. We were talking about that in lecture. It has an upper, middle, and lower division. That is your trapezius muscle. This is all trapezius, where you have number 1 arrow to the, to the neck area above that. That's trapezius. And below, to the tip of the arrow, you have another portion of muscle that is all trapezius. 2 is pointing to another muscle, very flat, very broad, called latissimus dorsi. Trapezius is a big muscle. That's why when you work your trap muscles, you have to do it in different directions. So trapezius and latissimus dorsi. Trapezius is one of those muscles. It's going to come back because it's innervated by one of the cranial nerves, accessory nerve. Okay, then here we have the chest. So again, on the side that has the number 1, that shows you the very superficial muscles, the layer of muscles you can see when we just take the skin off. On the other side, on this model's right side, or where you can see the 2 over here, that will be the layer of muscles deep to this number 1. So I have to peel off the top layer, and then you start to see the muscles below it. So those are what's called the deep muscle sides. Right? 1 is pointing to the big muscle of the chest called pectoralis major, and 2 is pointing to pectoralis minor. Now you can actually see where it's coming and going. Well, almost where it's going, but this is all pectoralis major at number 1. And you can see the clavicle to the right-hand side, then your sternum, and below that would be some of the ribs, or the cartilages from some of the ribs. And those are the 3 origins of pectoralis major. And you can't see what inserts on, on here, but it inserts on your humerus, on your, the proximal end of your humerus. Here we can see number 2, you have pec minor. And look, you can see where those white connections are here. It's attaching to these ribs. And then it inserts on the coracoid process of your scapula, or just the scapula. And then 3, we're looking at abdominal muscles. So number 1, pectoralis major. Number 2, pectoralis minor. And number 3, rectus abdominis. And then we have, I believe this is, maybe there's 2 more, maybe there's 1 more. Here we're looking at the abdominal muscle. So this is a one big torso, in the front of the room, one big model of a whole body. Right? And now it's now at least heads up and tail down kind of a thing. Right? You can see here to the left-hand side where the number 1 is, those are the superficial muscles. And over to the right, now you can see more depth. When you can see bones, you're deeper in it. Right? The muscles right through the center to the right and left of his umbilicus, right? That's rectus abdominis, straight up and in the middle, right? Originates on the pubic symphysis and inserts on, you can see ribs and a xiphoid process, right, of your sternum. But to the right and left are your oblique muscles, and you have an external oblique and an internal oblique. So the superficial side, those muscles that are traveling at a 45-degree angle, you can see their fibers traveling, from the waist area down and in toward your pubic symphysis. Right? That's the outer layer. So that would be the external oblique muscle. To the other side here at 2, clearly we're deeper inside. So that would be internal. Right? Again, muscle fibers are traveling in the opposite directions as number 1, but they're going essentially from your xiphoid process out and down toward your femoroacetabular joint, toward your hip. But they're still running at it like an oblique angle. So that would be the internal oblique muscle. External obliques, internal obliques. And then the last model is showing you the connective tissue layers associated with the muscle tissue. So this kind of coincides with those first, the first exercises are looking at the slides of muscle tissue, skeletal muscle, the dense regular connective tissue, which are the connective tissue layers, the epimysium, paramysium, myomysiums. That's what we're looking at here, connective tissue wrappings or layers. And then the third slide was the neuromuscular junction. Right? Well, this is letting you see in a model form what would be the epimysium in one. That's the connective tissue around the entire muscle and continues as the tendons. Then you have the next layer, which is the paramysium that's surrounding fascicles at number 2. And number 3 is the endomysium that surrounds one cell or one fiber. Remember, the endomysium is also seen on your other models. And these are the layers here, the descriptions of those layers. Epimysium, paramysium, endomysium. Okay, so that's the end of the images. I will see you on Thursday. Remember that any functions identified in the paperwork and the lab exercises that you filled out can be applied to all these models. All right, so I will see you then. Bye.